Good evening. Welcome, everyone. Thank you for joining us for this special event this evening. I'm Dr. David Dodick. I'm the board chair of the American Brain Foundation. The American Brain Foundation, as you know, funds research across the entire spectrum of brain diseases and disorders, including Alzheimer's disease and other dementias, Parkinson's disease, multiple sclerosis, ALS, stroke, migraine, and so much more. Because we know that many of these brain diseases are interconnected. So funding research across this broad spectrum of them, we believe, is our best hope for finding improved treatments, preventions, and ultimately cures. I believe that a discovery in one of these brain diseases or disorders will have a ripple effect on our understanding and treatment of others, which is why I believe so strongly in the Foundation's Cure One, Cure Many philosophy. And this is why I'm so excited for the opportunity to speak with and introduce you to some of the American Brain Foundation's next generation research grant recipients who are working today to discover advances in treatment and the cures of tomorrow. Joining us tonight are members of the 2023 class to share more about their incredible research projects. After they share, we will transition to a question and answer period where you all can ask our researchers any questions you'd like. Throughout the webinar, please feel free to put your questions in the chat, and our researchers will respond to you in the chat. But if it's a question that we believe would be better addressed verbally to the entire audience, they will save it. And we will get to as many questions as we can during the question and answer. I really hope you come away tonight with hope that a future without brain disease, which is our mission, is possible because of research projects like the ones you're going to be hearing about tonight. But first, let me tell you about the American Brain Foundation's Next Generation Research Grants Program. Through these grants, we fund and support innovative research by the best and brightest early career clinician scientists, including the ones you're going to hear from shortly. And over the years, this program has provided a total of $40 million to almost 300 researchers, most of whom, more than 85% of whom, have gone on to secure additional funding from the National Institutes of Health and other national funders. These grants are helping fund research, again, across the whole brain because of our belief that if we're going to find better treatments, prevention strategies, and cures, it's important to study the spectrum of these brain diseases. So we really want to thank our funding partners for making this program possible, including a special thanks to our founder and research partner, the American Academy of Neurology. Supporting early career researchers is important because, well, not the least of which, quite some time ago, I actually received one of these early career research awards. And it truly is what kickstarted my career, actually. It's what got me started. It's what led to additional funding. It's what launched my research career. And so it's just an absolute joy and a pleasure and a privilege to be able to come back full circle and see, because I would never have been able to compete with the people here tonight, I can guarantee you that. So I'm glad I went 30 years before you all. But it's just a privilege to be here tonight to come full circle and to be granting you all awards to kickstart your careers. This program really truly exemplifies the Foundation's mission of bringing donors and researchers together so that we can all one day live a life without brain disease. So without further ado, let's meet the researchers and hear about their work. And so first we're going to hear from Dr. Wesley Kerr from the University of Michigan, and he'll be sharing his research on epilepsy. Dr. Kerr, can you tell us a bit more about your project and how do you think this research will impact patients and provide hope for people living with epilepsy?
Thank you. I get the privilege of going first. So I'm Dr. Kerr. I'm at the University of Pittsburgh, actually. I moved a little bit. And I am both a seizure doctor and a biostatistician. So what I think about is how do I make data and I look at data in a way to address clinical problems. And one of the big clinical problems is how do we evaluate new treatments for seizures to say what is effective and what is not effective. We do a lot of trials and spend a lot of money doing trials. And I'm trying to think about how do I make these more efficient and more attractive to patients so that patients will be easier to be able to recruit and say, hey, I want to invest my time and my body in evaluating this new treatment. In specific, we have a problem in epilepsy that when we study uncontrolled seizures, if we give people placebo, which we need to give for sake of evaluating the new treatment, there's risks to placebo, including the risk of death on placebo that is higher than on effective medicines. So I really want to shorten the time that someone is exposed to placebo or any ineffective medicine. So what we're looking at is a lot of different trials and looking across a lot of different trials to say, are there ways that I can say early on in the trial, you're not going to respond to this medicine. I can stop the trial for you specifically after like a few weeks, 4 weeks, 5 weeks, maybe 6 weeks. But if you're responding very well to the treatment, we're going to keep on watching, keep on seeing that you're having that good response for the full length of the trial, which is usually at least 12 weeks. In that way, we're trying to make it so trials are safer and more attractive for patients to recruit. And that hopefully will help us develop new treatments and develop new cures. And I have the privilege of being supported by a lot of different people as part of this. My specific award is called the Susan Spencer Award. And Susan Spencer was a great clinician and researcher in epilepsy. This is also supported by the American Epilepsy Society, the Epilepsy Foundation, the Epilepsy Study Consortium, the American Academy of Neurology, and the American Brain Foundation. So there's a lot of people that I have the honor of being supported by.
Fantastic, Dr. Kerr. Thank you. Obviously, a really important study that you're doing. And I certainly have lots of questions, but I'm going to save them and move on to our next speaker. We'll come back to you, of course. Our next speaker is Dr. Sheena Baritono from Beth Israel Deaconess Harvard Medical Center, whose research focuses on cognitive aging and age-related memory loss. Dr. Baritono, floor is yours.
Hi, thank you. It's a pleasure to be here today, and I want to thank the American Brain Foundation, American Academy of Neurology, and the McKnight Foundation for their generosity in supporting this award. Since I got the award, I'm now an instructor and faculty member at Brigham and Women's Hospital, just down the street from Beth Israel at Harvard Medical School. So I'm an MD PhD, and as a physician scientist, I spend some of my time in clinic. And it was really, when I was going through training as a neurologist, I became interested in patients that developed the more atypical symptoms as they age. So something such as visual impairment rather than the more common symptoms like memory loss. These patients with visual processing dysfunction, they get into car accidents, they have falls, and they quickly lose their independence even if they have retained memory function. So it's important to identify these patients early to prevent bad outcomes. And hopefully with early diagnosis, we can do safety evaluations, cognitive rehabilitation training, and maybe down the line, some sort of brain stimulation, neuromodulation to treat these symptoms at their earlier stages when they would be more responsive. So I focus on brain connectivity and how brain atrophy causes dysfunction of brain networks and brain circuits. So you can simplify the complex organization of the brain. One way to do that is to think of a bunch of brain circuits like any electrical circuit. Different circuits are important for different functions and behaviors, and many of them are interconnected and therefore interact with each other. So I'm using brain atrophy, as seen on an MRI, as a way to kind of query and identify which of these brain circuits are altered in different behaviors. And my focus is on identifying the circuits that are important to visual spatial processing difficulties in aging. And I do that by looking at changes in connectivity due to brain atrophy. So I'm currently working with a large database of subjects, including healthy, unimpaired individuals, some with mild cognitive impairment, and some with Alzheimer's disease. And using the unimpaired individuals, using their brain scans or MRIs, I create a model to determine what the expected cortical thickness should be for every point in the brain. I then compare every test subject to that model to identify which areas are smaller than would be expected. And then I use those and define those as the atrophied areas for that subject. I then use a functional connectome to look at the brain circuits associated with those areas of atrophy. The functional connectome is built using special MRI scans from 1,000 individuals, And these MRI scans look at how different parts of the brain use oxygen. And we assume that the areas that use oxygen at the same time are working together and are therefore part of a brain circuit. So what I then do is I compare each person's atrophy circuit based on that connectome to their visual test scores, and therefore I can identify the circuits associated with visual impairment in patients that are, as they age, develop these problems. Once I identify these circuits, my plan is then to use them to see if I can predict which patients will develop visual processing impairment, so we can try to help them sooner and eventually, hopefully, identify targets for neurostimulation using these brain circuits.
Excellent, Dr. Baritano. Thank you. Really fascinating work. And we'll come back with some questions after everyone has spoken. Our next speaker is Dr. Dominique Popescu from Massachusetts General Hospital, who is doing research in neurodisparities. Dr. Popescu.
Hi, everyone, and thank you for being here with us tonight. It's really an honor to get to share my work with you. As mentioned, I am Dr. Dominique Popescu, and I work as a research fellow at Massachusetts General Hospital in the Department of Psychiatry. So at MGH, I work to understand the many causes of disparities in brain disease. So I'm particularly interested in disparities in dementia, in depression, and in stroke. Now, importantly, I want to mention here that a health disparity is a very special kind of specific thing that we're observing. And so a health disparity itself is a type of difference that's observed that is considered to be unjust, unfair, and unavoidable, and importantly, avoidable. And so I want to point this out here because that avoidable part is the actionable part. That's the part that I hope to give to patients with brain disease today. But right before I get there, I just want to describe what I'm actually looking at. So right now, what I'm looking at is we're seeing patients that are coming in specifically to MGH's stroke center. And we're seeing them come in at younger ages with more severe strokes. And we notice that those patients tend to be Black or Asian patients. But the very important thing that we noticed here is that there's something going on that's a little bit more than kind of just race. It's not Blackness, it's not Asianness, for lack of a better term, but there's something more. And I propose that that something more is social vulnerability. And the way we measure that is we look at your community that you live in, the air quality, the community level poverty, community level education. And I'm just really interested in how the environment, the characteristics of where we live impacts our brain health. So in all, my hope to give to patients living with brain disease right now is a better understanding of these causes, and in the future is something that we can do about it. So this research is informing a lot of other future research that's really focused on how to get services to people who need it, people with brain disease who have a social vulnerability, or in another project I'm looking at, psychological vulnerability. With all of that said, I want to thank the Edgar J. Kenton III MD Foundation, ASI, and of course the American Brain Foundation for this clinical research training scholarship in neurodisparities. Thank you very much.
Thank you, Dr. Popescu. Amazingly important research that you're doing. Can't wait to see the results. Our next speaker is Dr. Erica Williams from Mass General, Brigham in Boston. We got a Boston theme going here tonight. Her research is looking into the organization of the autonomic nervous system. So Dr. Williams, the floor is yours.
Thank you. Thank you very much for the introduction. And thank you also for the honor of being able to speak with all of you tonight. My research is focused on the autonomic nervous system, and to provide some context about what that is. We know the nervous system plays a really important role in detecting many external cues, right? So things like light, sound, touch, pain, temperature, right? These are all modalities that the peripheral nervous system plays a really important role in detecting and transmitting to the brain in order to regulate normal brain function. And we know that, for modalities like touch, pain, temperature, there are many conditions that can impact the function of those nerves and those neurons and cause them injury. Analogous to these systems that we have, these nervous systems that we have to detect external cues, we also have a nervous system or sets of nervous systems to detect internal cues. So these are sensory neurons that detect things like stretch in the intestine or chemical contents in the stomach or intestine. They detect stretch of the lung, and they detect the blood pressure in the major blood vessels that leave the heart. So all of the major internal organs receive kind of very dense and detailed innervation in order to detect these cues. And so we're increasingly appreciating that these internal senses play a critically important role. We know that they're dysregulated in many different forms of neuropathy and neurodegenerative disease, but we have very little understanding of the various neuron types that perform all of these essential functions in people, and we have very little understanding about how those neurons are changed in different disease states. And so in my work, I'm very grateful to the American Brain Foundation and the Foundation for Peripheral Neuropathy to allow this. We're going to be generating a map of these various different neuron types and the different types of machinery that they express and that they use in order to perform these critical functions. We're going to look at how those neuron types are affected in different disease states, even common things like poorly controlled diabetes or Parkinson's disease. And hopefully with this kind of molecular map of these different internal sensory neurons, we'll be able to approach autonomic disorders in a much more detailed and different way.
Excellent, Dr. Williams. Thank you so much.
Pleasure.
Next is Dr. Robert Hoyerman from Wash U in St. Louis. So we're making our way across the country now. We're in the Midwest. His research explores how dopamine affects pain signals in the brain and how that knowledge might lead to improved treatments for Parkinson's disease. Dr. Hoyerman.
Hi, everybody. Thank you so much for joining us tonight. So I am a movement disorders neurologist at Wash U in St. Louis, and the most common disorder that we see in our movement disorders clinic is patients with Parkinson's disease. Which we think of first and foremost as a movement disorder. Patients have tremors, slowness, stiffness, problems with walking and balance. But Parkinson's disease is really much more than just a movement disorder. Patients have a lot of other symptoms that we spend a lot of time discussing and addressing beyond their movement issues. Things like anxiety, they can have a lot of autonomic disorders to tie back in with Dr. Williams' work. And pain, in fact, is one of the most common non-motor symptoms that we see in patients with Parkinson's disease. And we really don't have a good understanding of why these patients are more susceptible to pain or sometimes experience spontaneous pain without any clear cause or trigger. So that's really where I'm starting with my research at a fairly basic level, starting by just looking in mice at areas in the brain that we know are responsible for pain perception and seeing how they respond to dopamine, how dopamine affects the signaling in these brain cells. And then working up to how changes in dopamine might affect pain behaviors in mice. And then eventually hoping to look at brain tissue of human patients who were gracious enough to donate their brains after they were done using them, and seeing if what we learned from mice might correlate back to the changes that we're seeing in human brains, with the goal of identifying better treatments for pain in these patients with Parkinson's disease. So huge thank you to the American Brain Foundation, as well as the AAN, the American Academy of Neurology, as well as the Parkinson Foundation, who were my co-sponsors for this grant and the opportunity to continue this research. And I was asked to briefly tie back into that core philosophy of the ABF of cure one, cure many. And I am not a pain researcher by background, so it has been incredibly humbling and educational for me to be in a pain research lab, learning from some of the foremost researchers in the field of pain biology and pain perception. And I am hopeful that what I might learn from this project looking at Parkinson's related pain will be translatable to other types of chronic pain, which, as many of you probably know, is a huge problem currently facing the country and the world at large, with limited treatments and opioids being not ideal and leading to a lot more problems than they sometimes solve. So that's a huge push in neuroscience in general to identify better treatments for pain. And I would love to be a small contribution to that effort.
Thanks, Dr. Hoyerman. It's a perfect example of this cure one, cure many philosophy. As you say, there's more than 50 million Americans in chronic pain right now, and so many other neurological disorders have chronic pain as a feature. And of course, chronic pain is a feature of many non-neurological disorders. So a perfect example of this cure one, cure many philosophy. Next up is Dr. Danwei Wu from Stanford University. We're on the West Coast now. So Dr. Wu, if you wouldn't mind sharing with us your research on potential treatments for multiple sclerosis, and actually how your research, again, connects the American Brain Foundation's Cure One, Cure Many model, and what kind of impact your research might have on other disease areas.
Of course. Well, good evening, everyone. My name is Dr. Danwei Wu. I'm a neuroimmunology fellow and postdoctoral researcher at Stanford University. As a neuroimmunologist, I'm really interested in pioneering new approaches to treat autoimmune disorders of the central nervous system. Our immune system plays such an essential role in fighting off infections and cancers, but under certain circumstances, our immune system can become hyperactivated, leading to damage of our own tissue. In a neurologic disorder such as multiple sclerosis or MS, the immune cells pathologically target myelin, which can in turn lead to permanent disability and neurodegeneration. To treat and hopefully one day cure MS, we need to understand how to restore our immune function. So to give a little bit of a background, our immune system is comprised of two main families of cells. One is myeloid cells. These are cells that give rise to macrophages and microglia. These are cells that can clean up debris and then present this debris to recruit other immune cells. And then the other main family are lymphocytes, which give rise to T cells, cells that can kill off infected or damaged cells, and B cells, which produce antibodies to guide these T cells. Essentially, hematopoietic stem cell transplantation is currently in clinical trials as a potential therapy for aggressive forms of MS. And the principle is that during hematopoietic stem cell transplantation, the bone marrow and the peripheral immune system is wiped out using chemotherapeutic agents. And this allows a new space for naive immune cells, either from the same individual or from a donor, to engraft in the treated bone marrow. And it's commonly believed that this introduction of the naive immune cells helps reset our immune system to a state where it can no longer recognize or target our own tissue, therefore treating autoimmunity. However, our lab also has demonstrated that bone marrow transplantation can also permanently affect myeloid cells in addition to lymphocytes, which was commonly thought to be the cells that cause MS. This is really significant because myeloid cells, which include microglia, are the most prevalent immune cells in the central nervous system. Unlike lymphocytes, the role of microglia in MS is not as well understood, but it's thought that the persistent activation of these microglia contributes to prolonged local inflammation and tissue damage. Microglia are also notoriously difficult to study and manipulate because they're protected by the blood-brain barrier. And unlike other immune cells that are from the bone marrow, they actually develop in early fetal development. And so we don't have the ability to manipulate the bone marrow as readily and affect the bone marrow microglia population. It actually turns out that during bone marrow transplantation, these chemotherapeutic agents non-specifically wipe out both lymphocytes and myeloid cells, including microglia. And what we found is that if you deplete the microglia using traditional chemotherapeutic agents, the bone marrow cells are able to migrate into the central nervous system and engraft, taking a permanent residence, dividing, and actually populating the brain. These cells, we call them circulation-derived myeloid cells. We're not really sure from current literature if these circulation-derived myeloid cells contribute to the clinical effect of bone marrow transplantation in MS, but that's what we're interested in studying. So what we've discovered preliminarily through our research is that these circulation-derived myeloid cells actually share features that are both similar and distinct with microglia. They both arise from the same cell lineage. They perform a lot of the same cellular function. But if you look at the genes that are expressed on circulation-derived myeloid cells, they actually are associated with homeostatic repair functions. We've also discovered that in a mouse model of chronic MS or EAE, these circulation-derived myeloid cells have anti-inflammatory effect that they can basically reduce the clinical severity in these mice and also reduce demyelination. We have also enhanced the infiltration of these myeloid cells by actually depleting the endogenous microglia further using a microglia targeting drug, in addition to looking at traditional bone marrow transplantation. And through this strategy, we can actually replace up to 90% of endogenous microglia in the central nervous system with these circulation-derived myeloid cells, enabling us to directly target microglia associated with disease states. So ultimately, my goal is to identify the precise signals that guide these cells into the central nervous system, control their engraftment, and maintain their long-term residency in the brain and spinal cord. I'm really excited about the potential therapeutic application of this research because our platform allows us to stably deliver therapeutic genes and treat potentially a various array of neurologic disorders. Actually, in our lab, we were looking at not just MS, but other neurologic disorders such as genetic leukodystrophies, lysosomal storage diseases, and as well as more common neurodegenerative diseases such as ALS and Alzheimer's. I believe that through this research, we can inform the development of novel cell-based therapeutic targets to treat a wide spectrum of neurologic disorders, very akin to the theme of the American Brain Foundation. And I really want to thank the support that I've received from the American Brain Foundation, as well as the National MS Society Clinician Scientist Development Award that's allowed me to do this research. And I'm really excited for the future of cell-based therapy. Thank you.
Thank you, Dr. Wu. Really elegant work. And again, a beautiful example of this cure one, cure many philosophy and how this work can have a ripple effect on the understanding of and the treatment of other neurological diseases. Terrific. Thank you. Next up is Dr. Paula Barreras from Cedars-Sinai. Her research aims to explore peripheral neuropathy. And so, Dr. Barreras, please tell us a little bit more about your peripheral neuropathy research.
Thank you. So, it's an honor to be here. I'm a neuroimmunologist by training, and my interest is inflammatory disorders of the nervous system, in particular a disorder called sarcoidosis, which can cause inflammation of any organ, including brain, spinal cord, and the peripheral nerves. A subtype of these nerves are called small fibers. These are the fibers that mediate pain sensation and temperature sensation. They are also involved in autonomic signaling, controlling, for example, blood pressure, heart rate, and intestinal motility. When these small fibers are damaged, we have a small fiber neuropathy. And the small fiber neuropathy can lead to chronic pain because these are the fibers that mediate pain. So these patients often describe feeling like their hands or feet are on fire, or it's very dramatic and can lead to severe disability and impairment in quality of life. In sarcoidosis, up to 40% of patients have a small fiber neuropathy. The mechanisms behind that small fiber neuropathy are not clear. While some people believe that it's the inflammation itself caused by the disease, there's no clear evidence of that. And other factors like diabetes, which is common in our patient population due to the chronic steroid use or exposure to toxic agents, can be independently the cause of the small fiber neuropathy. My project will try to clarify the role of inflammation in sarcoidosis-associated small fiber neuropathy. To do that, we're going to correlate symptoms of how severe and how advanced is the small fiber neuropathy in patients with sarcoidosis with markers of inflammation in the blood, called cytokines, and with markers of inflammation in the nerves and the skin itself. So we're going to look at skin biopsies that are usually done as part of clinical care, and we're going to stain those biopsies for specific immune cells to see if there is an infiltrate around these nerves. And in addition, we are going to correlate these clinical features, these cytokines, and these biopsies with outcomes in this patient population, so we can inform what predicts severe outcome or treatment response, and hopefully be able to inform patients about decision making in the future.
Thank you so much, Dr. Barreras. Really interesting work. Next up, Dr. Eva Kleinman from Washington University, again in St. Louis, whose research focuses on the underlying mechanisms that make the aging brain prone to neurodegeneration. Dr. Kleinman.
Hi, I wanted to start off by thanking everyone for giving me this opportunity to talk about my research, and especially the funding agencies that are responsible for making sure that I have the money to do my research. And that would be, of course, the American Brain Foundation, as well as the McKnight Brain Research Foundation and the American Academy of Neurology. So I am currently a movement disorders fellow at Washington University in St. Louis, and I am doing a 2-year research fellowship trying to get to the bottom, or at least start to discover what goes on in the human brain as it ages. So as opposed to a lot of what you've heard tonight about specific diseases, what I want to understand is what neurons in our brain are going through as we go from newborns to, healthy 20 and 30-year-olds on into individuals in their 70s, 80s, and 90s, and what that does to predispose us for diseases that develop as we age, things like Parkinson's and Alzheimer's disease. Traditionally, it's been a little bit hard to look at human brains as they age because people don't really want to donate brain tissue while they're still alive and using it. So the lab that I'm in actually has this really interesting way of using skin sample tissue, the way you just heard from Dr. Barreras, but instead of using the skin tissues and just fixing them and looking at them under the microscope, we actually keep the skin cells alive and can directly turn them into neurons. And what's cool about these neurons is that they reflect the age characteristics of the person who donated the skin sample. So if you take a skin sample from a 20-year-old individual and a skin sample from a 90-year-old individual, they're going to maintain a lot of the age characteristics even as you turn them into neurons. And so what I'm doing is I'm trying to figure out the different aspects of aging that contribute to neurodegenerative disease. And I am looking primarily at 2 things that help keep the neuron healthy and happy. And that is the cytoskeleton, which is sort of the scaffold behind a neuron holding its shape, and 2 different organelles inside the neuron that are responsible for helping maintain its health. Mitochondria, responsible for energy usage in the cell, and autophagosome, responsible for breaking down unused material and aggregates. And so my work is just focusing on, at this point, setting up what neurons look like as they age in terms of their cytoskeletal proteins and their organelle behaviors, with the hopes that down the line, I would then be able to try to compare those to skin samples from individuals either with disease or with prodromal disease who do not yet show whether they're going to develop something like Alzheimer's or Parkinson's, and be able to predict their risk of developing those diseases down the road.
Thank you so much, Dr. Kleinman. I think obviously this is research that's important to all of us. So fascinating work. Next up is Dr. Popely, whose research focuses on a particular type of dementia known as frontotemporal dementia. So Dr. Popely.
Thank you, Dr. Dodig. So I'd also like to begin by just thanking everyone here for the opportunity, honor. It's still very mind-boggling to me to think that there are people generous and gracious enough to fund the questions and ideas and kind of off-the-wall things that some of us are thinking about when it comes to brain diseases. And so my grant is funded particularly by the Holloway Family Foundation, Holloway Family Fund for the Association of All Frontotemporal Dementia. And I don't think this award would be possible without them. So thank you very much. I'm a cognitive neurologist. I'm interested in neurodegenerative disease. Similar to Dr. Baritono's excellent explanation, I think the way neurodegenerative diseases affect brain networks and brain circuits is a big piece of the puzzle in terms of determining who's at risk, how these changes happen, and eventually what do we do to manage those brain network and brain circuit changes. And so the types of frontotemporal dementia I'm really interested in are language predominant dementias. They're called primary progressive aphasia, and it's a rare form of dementia where people get changes in their language skills before they develop problems in memory or other thinking skills or behavior. And it can be very isolating. It's still a big question of why only the language circuits get affected in those people and not these other circuits until they progress later on. And so the work I'm doing is to look kind of two big purposes. One is, can we understand the brain networks using various forms of brain imaging, like functional MRI imaging or PET scan imaging, and define how these brain networks change with various types of language disturbance. And then the second part of it is, can we modify those brain networks using neuromodulation approaches? And so these are non-invasive stimulation approaches, things like a very small a microcurrent that travels through the scalp, through the skull, and to target a portion of the brain. And we're looking at a small portion of these patients with language disorders from dementia, and seeing if we can change some of those brain networks, change some of the connectivity, change some of the activity, and help us determine what are the best targets and what are the best types of modulations we can do to find those changes and hopefully lead to more symptomatic therapies that could be applicable to all sorts of dementia and all sorts of cognitive deficits beyond that.
Fantastic. Thank you, Dr. Popely. Really interesting and really important work. Finally, let's hear from Dr. Patricia Olson of Indiana University about her research into migraine.
Thank you, Dr. Dodick. Thank you so much for the invitation to join this amazing group of researchers. I've had a great time listening to my colleagues explain their research. I'd also like to thank the American Brain Foundation, the American Academy of Neurology, and Amgen for supporting my research. So as many know, unfortunately, but many know, migraine is a complex neurological condition. It's actually the second leading cause of debility worldwide. There currently is no cure for migraine, but there are treatments available, several treatments, in fact. So a challenge can become for providers and patients to find a medication or combination of medications that can help abort a migraine attack, decrease the migraine, the attack frequency, decrease overall debility, or improve quality of life. So many have heard of, I'm sure, the relatively new CGRP blocking agents. And they've really been a godsend for many. And since their discovery, it was found that some patients, it's life-changing. It's a miracle drug. Some patients are like, yeah, pretty good. I'm going to keep taking my other meds, but things are better. And some patients are just like, no difference. So for many patients with chronic migraine, it can take time. It can take months, even years, to come up with a treatment plan that works. So what if we were able to design a treatment plan that could spare the patient months or years of trial and error? So migraine itself is a multifactorial condition, but there is a strong genetic component in the form of migraine-associated variants. So under the mentorship of Dr. Will Renthal at the Graham Headache Center and Brigham and Women's Hospital, our group posed the question of whether there is a genetic component to treatment response as well. So again, what if treatment decision-making were informed by genetics? What if we could identify the response groups using biomarkers before having patients go through lengthy trials? So we hypothesized that migraine-associated variants could predict treatment response to these CGRP inhibitor medicines. So our study is a retrospective study first. So we're looking at a group of patients who are participants. They have migraine with aura or migraine without aura. They have donated their, I think it's their blood and their saliva to the Mass General Brigham Biobank. They have also allowed us to look into their clinical records, so into their clinic notes. And so we have those data of people who have that diagnosis, they're taking a CGRP inhibitor, and they have been genotyped. And we have their clinic notes. So our first step of this project then is to group participants based on their treatment response. Again, the ones with the life-changing effect, the ones who it's pretty okay effect response, and ones who just, it doesn't work at all. So we're developing a protocol and establishing parameters to extract data from the clinic notes using a human researcher, myself and another colleague, but then also utilizing machine learning to do the same. And we're comparing my data with then the machine learning data. So once we have grouped these participants, then we're going to look for genetic similarities among 3 groups. And what this has the potential then is to identify biomarkers for the CGRP response, with hope that we can then stratify options to guide individual therapies to treat migraine. Thank you again for allowing me to participate today.
Thank you, Dr. Olson. Really important work. I look forward to the results of that. Okay, so now we have an opportunity for everyone tuning in to ask any of our researchers any questions or anything that you might be curious about in the diseases that they're focusing on. And so please feel free to type your questions into the chat. And to get things started, I'm actually going to move right back to the top to Dr. Kerr. So your research focuses on shortening epilepsy clinical trials. Obviously very important for the reasons you mentioned. Tell us, how long is a typical clinical trial in epilepsy for presumably what are usually add-on therapies? They're already on a drug, you're adding another one. How long do those trials usually take? And how realistic are you that you can shorten them? And to what duration do you think you can shorten these to?
Very good questions. Thank you. A typical trial for medication-resistant epilepsy, which means they've already tried at least 2 medicines for seizure and they still have seizures, starts with like a baseline period where it's 1 to 2 months, figuring out how many seizures you have before we give you treatment. Then there's the starting the medicine phase. You build up to your goal dose. So that's the titration phase. And then there's that maintenance phase where you stay on drug for a period of time and see how you do. That whole process is usually 5 or 6 months at least. And we're really focusing on that maintenance phase. How do we shorten that? Because the other things we sort of need for other reasons. I mentioned that maintenance phase is usually 12 weeks long. What we're seeing across the 11 trials that we're analyzing is that on placebo, you're really only in the placebo arm for like a month and a half. For people that particularly have a poor response, you can be in placebo for even 2 weeks, and that's fine. Mm-hmm. So for the people who really are not responding and continue to have seizures, don't have any effect of that treatment, we're really shortening it. But for some people who are having great responses, like they're seizure-free for 2 or 3 months, this is relatively safe to keep you in the trial, even if you're on placebo, because if the placebo is working, I'm totally okay with that. So this differential where we want to keep people safe if they're having more seizures, and also keep people safe in the trial if they're responding, meets this sort of dual goals of trying to evaluate our new treatment and also respect the patients who are using their bodies to help evaluate this treatment.
Yeah, thank you. And just by the way, what's the proportion of people who have already failed 2 anti-epileptics are likely to respond to a third new agent?
Very low. So usually about 2/3 of people will respond to the first or second appropriately chosen medicine. And once you get to the third or more, you maybe have like a 3 to 5% chance of seizure freedom on medicines. So that is a very low chance. Part of what we're trying to do with new treatments is to improve that chance of response and also improve that chance of response to the first medicine and the second medicine. To swing back to the treat one, treat all, this concept of identifying treatment response earlier and predicting future treatment response you heard of in the migraine talk. So these sorts of statistical techniques, we develop them in epilepsy and they go other places too.
Terrific, thank you. Coming to Dr. Baratono, your research focuses on visual spatial dysfunction in people who are aging, with cognitive aging. So I guess one of my questions is, can you assess visual spatial dysfunction before a patient, when they're in the preclinical phase of a neurodegenerative disease, let's say? Should we be doing that clinically? And then how would you intervene from a visual spatial standpoint to actually improve visual spatial function?
Great question. So we do, in a healthy individual, we don't really test cognitive function really at all until somebody presents with difficulties. And oftentimes patients don't realize there's an issue necessarily at those stages either, especially if they end up having something like Alzheimer's disease pathology in the long run, where they kind of lose some of that insight sometimes. So I do think it would be worth doing a little bit more visual spatial processing, especially if you ever have a patient that had a fall because they didn't see something, or they got in a car accident, something where you start to get that hint that the visual processing system might be impaired on some level. Then that might be somebody that should get more in-depth testing. The other thing I'm hoping to do is to predict these patients based on the MRIs. A lot of people get MRIs or CT scans because of a fall or for something else, like they had headaches. So there's other reasons why people go in to get these brain scans. So if we can have some sort of automated system where we can process these scans and say, hey, this person is at risk for this deficit, then maybe we can try to take those patients and do more processing with them. And in terms of, what can we intervene on? What can we do? So there's some data that cognitive rehabilitation can be helpful for visual spatial dysfunction. Cognitive rehabilitation is it's a vague term I'm using because different places do it differently, but it can involve training with different types of visual stimuli. But I think the other thing that would be great, and this kind of gets to what Dr. Popley was talking about, would be neurostimulation. And can we bring these patients in earlier, and we can use either transcranial magnetic stimulation or direct current stimulation to try to help boost the strength or the healthy circuits, or maybe even recruit some of the other circuits to kind of take over and help with those functions. So I think that would be a wonderful application eventually for these types of research projects.
Terrific. Thank you so much. Dr. Popescu, your research on healthcare disparities and neurodisparities specifically, and in stroke patients in particular, is the goal ultimately to identify those vulnerable populations, where they are, and to improve access to care and services for them, particularly with respect to the delivery of acute stroke care? Is that the ultimate goal?
Thank you for your question. Yeah, the ultimate goal of understanding these components that are impacting stroke severity is that while access is always an important issue, what we're starting to see out of the first kind of health disparity waves of research is one of the hypotheses was, hey, folks may not know that they have a higher risk based on these social aspects that they're living with. And it's starting to look like actually folks are pretty aware. So it's not, and it may not necessarily only be an access issue for some of these communities. So while access is always important, and all of the research efforts that are driving increasing access and things like that are incredibly valuable. I think it's specific to the location here. So I'm specifically talking about Massachusetts here in the kind of greater Boston area. I think what we're really seeing here is that there's something we need an intervention point that's a little bit earlier, that's a little bit sooner. And that's really tricky. That's the part that I'm trying to understand, getting to work with colleagues who are neurologists and how we interact with these patients, because the heart of this is it seems like folks are getting missed. It seems like a life course view would have been, in a perfect world, a life course view would be phenomenal to give services to these folks before they're presenting at a younger age with more severe strokes and such. So, but to narrow down to the not perfect world that we live in, what I think the power in understanding the contribution of these social factors to things like stroke severity is understanding which patients are more likely to benefit from additional support after we've gotten the post 90 days of that big event, the stroke. Once we get 90 days post, there are a lot of after effects, a lot of downstream effects that we see that could still be driven very much by these social factors. So when somebody has a stroke, for example, and they also go on to develop depression, which does happen to a large proportion of stroke survivors. Now imagine being in that situation. You just survived you survived the stroke. Now you've also developed depression, and now your long-term outcomes are you are at risk for worse long-term outcomes. Short answer, things are harder. So we're hoping that with this information, we can identify those folks who excellent neurology has gotten them through to that 90 days. What more can we do to support even more continued recovery?
Terrific. Thank you so much. And thank you for your work. On to Dr. Williams. You mentioned your research focuses on mapping different neuron types in the autonomic nervous system and how they may be preferentially affected in different neurological diseases. For our audience, can you talk to us about some of the diseases where the autonomic nervous system is dysregulated? So many of our audience members may not be familiar with the autonomic nervous system and how it expresses itself clinically.
Yes, excellent question. So the autonomic nervous system plays an important role in regulating all of these internal physiologic functions. So when it's not working well, patients will have symptoms like a lot of difficulty moving from a lying to a standing position, for example, because in order to do that, your body has to augment the blood flow to the brain to accommodate the effects of gravity. And the autonomic nervous system plays a critical role in being able to do that. Autonomic nervous system also plays a really important role in digestion and gastrointestinal motility and kind of the movement of food and digested food contents through the various stages of digestion in the gastrointestinal tract. So how does that manifest for a patient? Some patients can have very early satiety, difficulty eating a full meal. You can have tremendous nausea or discomfort with eating, or they can have a lot of constipation or diarrhea, depending upon the region of the GI tract that's impacted. Other neurons play an important role in the regulation of breathing, and so patients can have sensations of dyspnea or shortness of breath, and kind of out of proportion to what they would normally feel with that level of exertion. And so the autonomic nervous system is very diverse and plays an important role in many different physiological states and sensations. And so people can present with a broad diversity of different kinds of symptoms when this nervous system is impacted. And so the specific diseases that are impacted by that impact the autonomic nervous system are also similarly varied. One of the most common that we see, particularly in the neuromuscular clinic, are diseases with other forms of neuropathy in which the autonomic nervous system is also impacted. So a common one is actually diabetes. A very large proportion of patients with diabetes develop injury not just to the nerves that go to the feet and the legs and the hands, but also to this autonomic nervous system. And so they have a lot of difficulty with GI motility, and they also have a lot of orthostatic intolerance, the name for this inability to change positions from lying to standing or sitting to standing without getting very lightheaded. Similarly, I mean, as some of our other researchers have mentioned, Parkinson's disease actually comes with a very broad set of autonomic symptoms. And there's a hypothesis that is still undergoing a lot of investigation, but there's an idea that Parkinson's actually starts in the periphery and travels centrally via autonomic nerves. And there's some data on population-based studies that when those tracts are disrupted, looking at these very large populations, the risk of Parkinson's disease isn't fully eliminated, but it's reduced. There's some anecdotal, like similarly. So diabetes, Parkinson's disease, Guillain-Barré, other types of autoimmune neuropathies, metabolic neuropathies, toxic neuropathies, and other forms of neurodegenerative disease impact this peripheral autonomic circuitry and can have really devastating impacts on quality of life for patients with those conditions.
Thank you. Thank you very much. Dr. Barrera, as you talked about your research focusing on small fiber neuropathy and all of the clinical symptomatology that can come with that as a result of inflammation, particularly with respect to sarcoidosis. I guess I would ask, why did you focus on sarcoidosis, which is a relatively rare disease, and what other diseases can cause small fiber neuropathy as a result of inflammation?
Thank you for your question. Well, the first, why sarcoidosis, I think, is both a fascinating disorder and a disorder that we have yet to learn much about. So this disease affects many people, particularly those of African descent and a lot of vulnerable populations. It can affect any organ in the body, and yet we don't really understand the cause of sarcoidosis. We don't have a lot of tools to understand the prognosis of the disease, and specifically in those patients with sarcoidosis and small fiber neuropathy, there's very little research done. So I wanted to focus in an area where I could make an impact, where what I find is informative for the patients with that disease and potentially other similar disorders. Now, what other diseases can cause a small fiber neuropathy? Diabetes is a big one. As mentioned before, people that live with diabetes can experience damage to autonomic nerve fibers, and these are small fibers in the periphery leading to numbness and pain. But also other immune-mediated disorders like Sjogren's disease, for example, can lead to small fiber neuropathy. So the interesting question here is sarcoidosis responsible for the small fiber neuropathy that we're seeing in these patients? It may be that it's not, that is due to comorbidities like coexistent diabetes. And the reason that question is important is because treatments that are immune-targeted, immunosuppressive treatments, are not without harm, right? Like, so we don't want to use them if we don't need to. And if they work, we want to use them when we need to. And currently, when people show up in a clinic with sarcoidosis and small fiber neuropathy, we don't really have clear guidelines of what to do. So you'll see neurologists try to treat exacerbations of pain with immune suppressions, and in some other clinics, they'll say, no, I'll just observe you and perhaps give you some pain medicine. And we really need data to drive those decisions. So that's what I want to do. Also, I want to make sure I mention that I want to thank the American Brain Foundation and American Academy of Neurology and the Foundation for Peripheral Neuropathy for funding my research, since I didn't say it before. Thank you all.
Thank you so much, Dr. Barreras. If we could squeeze in like two more quick questions, we will have, I think, gotten to everybody, because some of our researchers have been answering questions in the chat. So Dr. Wu, quick question. Your cell-based therapy is a possible treatment for MS. Could this lead to breakthroughs for people with diseases that lead other neurological diseases?
Yeah, I think we're like, I know a lot of the studies I'm doing are in mice and preclinical models, but bone marrow transplantation, I think, has been explored for a lot of other different types of neurologic disorders already that are not necessarily immune mediated. And the ones I commonly think about are like genetic leukodystrophies. I believe for metachromic leukodystrophy, there is a phase 1, phase 2 trial that was published in 2022 where they did bone marrow transplantation on genetically modified hematopoietic stem cells to replace the missing enzyme in that disorder. And to be fair, there's a lot of genetic white matter disease that they've kind of tried with bone marrow transplantation, have seen favorable results. And they don't really understand the exact mechanism. We do know that a lot of neurologic disorders that involve the central nervous system also cause disorders are related to disordered microglia. There are diseases that are primary, genetic disorders where there's the function of microglia is impaired. And then there's also disorders in like Alzheimer's or ALS where we see microglia that are overactivated. And so I think the first, target, I think that would be like I don't want to say a low-hanging fruit, but would be an obvious target would be for these genetic leukodystrophies, which unfortunately are fatal. in a lot of conditions, and they're both pediatric and adult onset. And I think this is a tool that we can utilize in that context to treat another type of white matter disorder separate from MS. And then I think, this could be refined to treat other more common disorders, possibly related to aging. So I think it's probably closer on the horizon than most of us think, because it's, a lot of it's still in preclinical models, but we're already applying a lot of these same strategies for treatment in rare genetic disorders.
Thank you, Dr. Wu. Appreciate that. Finally, I think we'll end with Dr. Hoyerman. And I guess my question, Dr. Hoyerman, is most people don't associate chronic pain with Parkinson's disease. So how common does that happen? Number one. And number two, has dopamine been implicated in chronic pain with other disorders, be they neurological or otherwise?
Yes, those are very good questions. Chronic pain was not something that I thought about in relation to Parkinson's disease, really until I kind of serendipitously landed in a pain lab and was looking to develop a project to combine those two interests. So the studies vary in terms of how common it is, anywhere from 50% to 80% of patients with Parkinson's disease will have some form of chronic pain. That's a little bit confounded by it being an older population, and many people as we age will have low back pains, other types of pains, but consistently is higher than age-matched populations. And there's also been several studies showing directly that there's an increased sensitivity to painful stimuli when they directly test it. So is something that is very common and is, I agree, not a well-recognized association. I think from that standpoint is ripe to ask these questions and better understand why that might be the case, and may lead to better understanding of pain in general. There is more and more that is being learned about how dopamine impacts pain sensation, really across the whole pathway of pain sensation. So there's descending dopamine signals that affect how pain is processed at the spinal cord level, really all the way up through the areas of the brain that are responsible for processing pain. And so we're, I think, in the early stages of starting to piece together how dopamine really impacts all those various areas and how it might be used therapeutically. But thus far, there are no dopaminergic medications that are used specifically for chronic pain.
Great. Thank you so much. Maybe one more question. I'll squeeze one more in, Dr. Olson. Have you identified, first of all, what proportion of people out there with migraine actually respond to these CGRP-based therapies? Because you mentioned some don't respond at all, some respond really well, and some respond, partially. So my question is, what proportion respond really well, number one? And number two, have you identified anything in your practice that helps you identify, at least now, potentially the likelihood that someone's going to respond or not?
Dr. Dodek, I don't have a satisfactory answer for you for that first question, because there's the CGRP, the monoclonal antibodies, injectables, as you know, but they're the ones that people can inject once a month or once every 3 months for preventative, or they can take it the galcanezumab or erinumab or fremanezumab, or they can take the ubrogepant or rimegepant as needed. So I want to say, gosh, I don't even want to give you a quote, because I did look this up. When also the reason why I don't have a satisfactory answer for you, because the treatment response definition is different based on the different studies.
Yeah.
And so, for example, and that's what we found in our project too, is how do you define treatment response?
Right.
So, so there are, I don't want to say that it's 30%, because you have a responder, but then you also have a super responder.
Mhm.
And so I don't have the exact percentages for you, but I can say that that's where we ran into our issues with even defining treatment response in these clinical notes, because I'm reading a note as a provider and as a researcher, I'm reading other providers' notes. And it's been peculiar and very interesting to pull objective data from subjective text.
Yeah.
That is doing much better.
Yeah.
Continue the medicine. And the goal with this project is to determine whether or not, and I didn't speak in depth about this further, but is to determine whether or not my impression of the response can match a machine learning impression response. So we're teaching then the AI of how to pull just treatment response.
Yeah.
I can tell you that in my own practice, it surprises me, and it's taught me a lot about my own personal inferences that I take from patients, because I'm thinking, just as I'm saying, how many headache days difference, how many migraine days difference, and I'm talking, and I'm in my mind, I'm thinking, oh, they're going to want to change. And so then I say, well, what do you want to do? And I don't guess well. I have to confess to you, because sometimes they're like, oh yeah, keep it going. And I'm thinking, really? And that's why there's that quality of life piece that is so hard to quantify or improved ability. So what I can tell you that for our project, we've assessed a treatment response in order to group these three into headache days, migraine days, and then the global impression of change.
Okay, that's good. That's good. It was a tough question, but I appreciate that response. Clearly, it's important because the medicines are expensive, and once you give one of these injections, it sticks around for 5 months because of the long half-life. So it's really important question. I think I better stop, or I'm going to get thrown off the stage here. So first of all, I can't thank you all enough for the work that you're doing, for showing up here tonight, for speaking with our audience here. Incredibly important work that each and every one of you are doing. And I look forward to seeing the results over the next year or two, and look forward to following the progress of your career, truly. Also, I want to thank the research partners. Many of you have mentioned that there are many foundations and organizations that have funded your work. And so we rely heavily on these research partners who provide 2/3 of the funds needed to fund your research. And so we rely on their generosity to make this happen. And we also rely on the generosity of individual donors who have been very generous in providing funding to the American Brain Foundation, which we turn directly over to researchers like you. So for those of you listening tonight, please donate to the American Brain Foundation so we can continue to support incredible researchers like you've heard tonight. Every dollar toward research counts. So I want to thank you all very much for coming tonight. I'm sorry to keep you late. I could keep this going for another couple of hours, but thank you all again for being here tonight.